So last time we'd gotten as far as talking about junctions, and we'd talked about anchoring junctions, the desmosomes, the hemidesmosomes. We're going to finish that story about junctions today, and we're going to start with the idea of a gap junction. The gap junction is a kind of communicating junction. The gap junction is an intercellular junction. It connects adjacent cells. And so what we're going to find is that these junctions are made of transmembrane proteins. Now, a gap junction will contain maybe as many as 100 or 200 connexons. Each connexon is a channel, but each connexon is composed of 6 connexins. Connexins being the individual proteins. And we'll show how these individual connexins go across the phospholipid bilayer, how they then form a connexon, how the connexons come together to form a gap junction. The function of these junctions is to allow the passage of small molecules from one adjacent cell to the next. They will allow the passage of any molecule less than a molecular weight of about 1,000. So if you start thinking about this, then you begin to recognize that the kinds of components that will pass from cell to cell will be things like ATP, ions. Those molecules that have molecular weight less than 1,000 will easily pass through these channels. And so we'll talk a little bit about that. We'll spend a moment on tight junctions. Tight junctions are actually zippering the membranes of adjacent cells together. The tight junctions, though, are not an especially strong junction. They're basically just proteins embedded within the phospholipid bilayer. But what they do is they provide a blockade or an occlusion between the spaces that separate adjacent cells, so that molecules from the lumen of whatever tube we're lining don't easily pass between the cells and down into the underlying tissues. And so we'll just show how these are functioning. And then we're going to talk about a modified form of epithelium. We'll modify epithelium into something called a glandular epithelium. And we'll talk about the glandular epithelium as being a component of exocrine glands. So in the next few days, we're going to talk about exocrine glands and endocrine glands. This story today is exocrine glands. Exocrine glands are composed of an epithelium. Because they're composed of an epithelium, and the definition of an epithelium is to be continuous to the outside world, then we're going to see that these exocrine glands are now going to dump their products out into the extracellular space. And, well, I should say out into a surface. So, for example, if you imagine a sweat gland or a mammary gland, all of those components, all of the secretions of those glands go out onto the surface. Even if you're thinking about the pancreas, which is another exocrine gland, the digestive enzymes will be secreted from epithelial cells in the pancreas, go out into the lumen of the small intestine, and then the lumen of the small intestine will extend to the outside world. So you can walk your way from the secretory epithelium of any of these exocrine glands out onto the surface. If we're talking about sweat glands or sebaceous glands or mammary glands, their products easily go out onto the surface. But it's even true for organs which are buried as deeply as the pancreas, where these exocrine glands are releasing molecules to the outside. And we'll talk a little bit about that. And then what we'll try to do when we finish the story on epithelium is we'll then spend a little bit of time talking about bringing together two kinds of tissues. Whenever you combine tissues, you generate an organ. The simplest of all the organs is skin. And skin is considered to be an organ because it is composed of an epidermis, produced from stratified epithelium. The epithelial cells here will be keratinocytes. So again, you'll harken back to what we talked about the other day, where the keratinocytes are cells that are rich in keratin, the intermediate filament protein, the cytoplasmic filament protein that binds to the plaques and holds the desmosomes and hemidesmosomes in place for these skin cells. And then we'll find the epidermis, which is an epithelium, sits on a connective tissue layer, which is called the dermis. And the dermis is formed from fibrocytes spinning out an extracellular protein matrix. we'll see again that the dermis is well vascularized. There's a lot of blood that goes to the dermis. That's why if you cut yourself, it's a very shallow cut and only goes into the epidermis, you don't bleed, because there's no blood going into the epithelium. But there's quite a lot of blood that goes to the dermis, the connective tissue layer. And we'll talk a little bit about that. And then we'll spend a little time talking about melanocytes. Melanocytes are cells which sit at the interface between the epidermis and the dermis. These cells are not really connective tissue and they're not really epithelial tissue. They actually originate from something known as the neural crest. In development, they migrate out as part of the neural crest, and the neural crest becomes a whole bunch of things in the periphery, and one of them is the melanocytes. What the melanocytes do is they secrete a pigment. And we'll talk a little bit about this because it's the secretion of the pigment which is then taken up by the keratinocytes that protects the dividing cells of the epithelium from ultraviolet light. And we'll spend a little time talking about that. And then if there's time, we'll get into appendages of skin, which will be hair, glands, and nails. And these will be extensions of the epithelium. And we'll talk about how hair is really just a different kind of keratinocyte. And in fact, our toenails and fingernails are just a different kind of keratinocyte. And if you extend that to animals and look at the hooves of horses or cattle or pigs, those hooves, in fact, are just a kind of nail. They're a kind of keratinocyte.